Hello everyone, welcome to my YouTube channel. Today we are going to discuss about printing of bone. So printing of bone, as we discussed about printing of ear yesterday, in the previous class. So printing of bone we are going to discuss in this particular class. So printing of bone in itself, it is nothing but what? Again, bioprinting of three-dimensional structure of a bone. Now, for example, why we have to print the bone? Because most of the times a bone, either it is not developed properly, or whether the bone has gone deformed because of some genetic issues or some other environmental factors, or it may be because of the accidents the person loses his leg or hand. That's why we need to make sure that he should get his bone back. So for that, those kind of applications or the situations, we go for the 3D printing of bone and make sure that this particular product should follow this particular functions of the bone. It should act as a normal bone so that we can replace the deformed bone, or the person who is not who has lost his particular leg or any other parts of the body, so where the bone is involved, that can be implanted. Even for example, sometimes the people lose because of the accidents and all, their even fingers will be ruptured, so we can have a 3D printer of this thing and we can replace with that. Okay, these all things comes under the situation where we require a 3D printing of bone. So now 3D printing has revolutionized the field of medicine, right? Definitely one of its application is 3D printing of bones. So this process involves using a 3D printer to create bone shaped structure using a specific material such as biocompatible polymers, ceramic materials as ink. So that means what they are trying to say is 3D printing is nothing but why we use here, for example, to create the bone, and for what materials it contains? Biocompatible polymer materials, ceramic materials as a ink. So the ceramic material is very important when you are going for the printing of bone. Okay, so along with the biocompatible polymer, there will be a ceramic material also, and most of the times we will be using to create as a ink. Ink means what we want to print as a composition of a bone. So now the printed bone structure can be then implanted into the patient to replace missing or damaged bone tissue. As I told you, if this particular part of the the finger is missing or damaged, you make that particular 3D structure, you fix it here, that's it. Okay, so there are two main approaches for the 3D printing of bones. One is additive manufacturing, scaffold based techniques. ಸೊ ಅರ್ಥ ಇದಲ್ಲ. ಸೊ ಇಲ್ಲಿ 3D ಪ್ರಿಂಟಿಂಗ್ ಆಫ್ ಬೋನ್ ಅಂದರೆ ನಮ್ಗೆ ಎಲ್ಲೆಲ್ಲ ಬೋನ್ ಎಲ್ಲ ಬೇಕು, ಆಕ್ಸಿಡೆಂಟ್ ಆಗಿ ಏನಾದರೂ ಆಗಿರ್ಬೋದು, ಅಥವಾ ಡಿಫಾರ್ಮ್ ಆಗಿ ಹುಟ್ಟಿದಾಗಿಂದಲೇ ಏನಾದರೂ ಬೋನ್ ಮಿಸ್ಸಿಂಗ್ ಇರ್ಬೋದು, ಅದನ್ನೆಲ್ಲ ರಿಪ್ಲೇಸ್ ಮಾಡೋಕ್ಕೆ ನಾವು ಬೋನನ್ನು ಪ್ರಿಂಟ್ ಮಾಡಬೇಕಾಗತ್ತೆ. ಅದಕ್ಕೆ ಬೇಕಾಗಿರೋ ಮಟೀರಿಯಲ್ಸ್ ಏನಂದ್ರೆ ಬಯೋಕಂಪ್ಯಾಟಿಬಲ್ ಪಾಲಿಮರ್ಸ್, ಸಿರಾಮಿಕ್ ಮಟೀರಿಯಲ್ಸ್ ಆಸ್ ಅ ಇಂಕ್ ಆಗಿ ಯೂಸ್ ಮಾಡ್ತೀವಿ. ಸೊ ಆ ಇಂಕ್ ಅನ್ನ ನಾವು ನಮ್ಗೆ ಗೊತ್ತಲ್ಲ, ಜನರಲ್ ಅಪ್ರೋಚ್ ನಾವು ಸ್ಟಡಿ ಮಾಡಿದ್ದೀವಿ. ಇಂಕ್ ಅನ್ನ ಹಾಕಿ ಮತ್ತೆ ಅದನ್ನ ಪ್ರಿಂಟ್ ಮಾಡಿ ನಾವು ಅದನ್ನ ಫೈನಲಿ ಒಂದು ಆರ್ಗನ್ ಆಗಿ ಕ್ರಿಯೇಟ್ ಮಾಡ್ತೀವಿ. ಇದರಲ್ಲಿ ಎರಡು ಸ್ಟೆಪ್ ಇದೆ, ಎರಡು ಪ್ರೊಸೆಸ್ ಇದೆ. ಓಕೆ, ಟೈಪ್ಸ್ ಇದೆ, ಅಪ್ರೋಚ್ ಇದೆ. ಒಂದು ಅಡಿಟಿವ್ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರಿಂಗ್ ಮಾಡಬಹುದು ಅಥವಾ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಬೇಸ್ಡ್ ಟೆಕ್ನಿಕ್ಸ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಬಹುದು. ಸೊ ಅಡಿಟಿವ್ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರಿಂಗ್ ಅಂಡ್ ಆಸ್ ವೆಲ್ ಆಸ್ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಬೇಸ್ಡ್ ಟೆಕ್ನಿಕ್ಸ್ ಕ್ಯಾನ್ ಬಿ ಯೂಸ್ಡ್. ಸೊ ಅಡಿಟಿವ್ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರಿಂಗ್ ಇನ್ವಾಲ್ವ್ಸ್ building up the bone structure layer by layer. So layer by layer you are building up the bone structure. That is what it is called as additive. Okay, so whereas scaffold based techniques involves creating a porous structures that provides a framework for the bone cells to grow and development. So understanding, in the first structure, that is additive manufacture, we are creating layer by layer bone structure. Now bone structure is cleared. Now in second approach, what happens? We are going to create a porous structure. Okay, why we need this porous structure? It will provide a framework for the bone cells to grow and to develop. So that this particular capillaries, or you can say the pores, so that it will give all the necessary nutrients, proteins or supply, so that the bones will start growing it. ಓಕೆ, ಸೊ ನಮ್ಗೆ ಇಲ್ಲಿ ಎರಡಿದೆ. ಅಡಿಟಿವ್ ಅಂದ್ರೆ ಲೇಯರ್ ಬೈ ಲೇಯರ್ ಅನ್ನ ಬೋನ್ ಅನ್ನ ಫಾರ್ಮ್ ಮಾಡ್ತೀವಿ. ಎರಡನೇದು ಏನಪ್ಪಾ ಅಂತಂದ್ರೆ ಅದು ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಬೇಸ್ಡ್ ಟೆಕ್ನಿಕ್. ಅದರಲ್ಲಿ ನಾವು ಎಲ್ಲೆಲ್ಲ ಪೋರ್ಸ್ ಬೇಕು, ಎಲ್ಲೆಲ್ಲ ಹೋಲ್ಸ್ ಬೇಕು ಅಲ್ಲಿ, ಅಥವಾ ಬ್ಲಡ್ ಅದಕ್ಕೆ ಏನಾದರೂ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಸಪ್ಲೈ ಎಲ್ಲ ಬೇಕು ಬೋನ್ಸ್ ಅಲ್ಲಿ ಗ್ರೋತ್ ಆಗಲಿಕ್ಕೆ, ಅದಕ್ಕೆ ಯೂಸ್ ಆಗ್ತದೆ. ಸೊ ದಟ್ ವಾಸ್ ಅಬೌಟ್ ಸೆಕೆಂಡ್ ಅಪ್ರೋಚ್. ದ ಫಸ್ಟ್ ಅಪ್ರೋಚ್ ವಿ ವಿಲ್ ಸ್ಟಡಿ. So first approach, additive manufacturing involves building up the bone structure layer by layer using the biocompatible materials. This sentence is repeating what we are doing. Additive manufacturing means layer by layer we are building the bone. Now the bone has to be obviously with the biocompatible materials. So layer by layer deposition of the material enables what it is enables it creation of complex three-dimensional structures that mimic the nature of the bone, because our bone itself is a complex structure. So it is also made by layer. So that's why we are layer by layer we are making, so that we can have the complex three-dimensional proper structure. So now the process of additive manufacturing in 3D printing of bone involves several key steps. So those steps we will discuss. So these are the major steps. We are having around 8 steps. The first step is patient imaging. Generally I will explain once and then we can go in detail. First, patient imaging. You need to make sure that you are taking the patient image either by using CT scan or MRI scan. Next, digital model generation. You need to make a three-dimensional model by using the imaging data, so that you are getting the data from CT scan, MRI scan, so that a three-dimensional structure design you have, that design you need to prepare. Third thing is scaffold design. So now the scaffold, to how exactly that design is there. Now to do that, the scaffold also three-dimensional structure you need to prepare, so that in this particular you can have the different appropriate size, the shape and internal structure that can be modified. If the size of the ear is big, that bigger scaffold can be designed. If the size should be less, that can be done. And shape, it depends on if it is ear, different shape. If it is liver, it is different shape based on that. And then internal structure of the scaffold to match the patient's anatomy. Okay, all those things can be specific requirements. Those things can be designed. So that is called a scaffold design. Okay, you can use some software so that you can have make sure that there is a proper support, porosity and structural integrity in the scaffolds. Then the fourth step is material selection. Now we need to select the material. Material selection, yesterday we discussed the same copy paste here. So what we need for our material to be selected for this 3D printing, since we are already using it for the humans, it should be biocompatible. Okay, make sure that you are chosen for this particular 3D printing process, whatever the materiality, it should be biocompatible. That means it should not create any adverse effects to any other cells, okay, after implantation. So these materials should be capable of supporting the cell attachment, growth and eventually bone regeneration. So this particular material, whatever biocompatible materials you are using, that should do 3 functions. It should support cell attachment. From cell to another cell, attachment should be there. That also it should support. Cell growth it should support, that so that cell can grow, and eventually bone regeneration. Finally, you will get the bone regenerated. So common materials include biocompatible polymers, ceramics, composites or biodegradable materials. ಅರ್ಧದಲ್ಲ ಫಸ್ಟ್ ಪೇಷಂಟ್ ಇಮೇಜ್ ಮಾಡ್ತಾ ಇದ್ದೇವೆ, ಅದನ್ನ ನೀವು ನಾರ್ಮಲ್ ಪೇಷಂಟ್ ತಗೊಂಡೋಗಿ ಹಾಕಿ ಅದರ CT ಸ್ಕ್ಯಾನ್ ತಗೊಳ್ಳಿ, ಅಲ್ಲಿ ನಿಮಗೆ ಇಮೇಜ್ ಸಿಗುತ್ತೆ. ಆ ಇಮೇಜ್ ಇಂದ ಸೆಕೆಂಡ್ ಸ್ಟೆಪ್ ಡಿಜಿಟಲ್ ಮಾಡೆಲ್ ಜನರೇಷನ್ ಮಾಡ್ತಾ ಇದ್ದೇವೆ, ಅಂದ್ರೆ 3D ಡಿಸೈನ್ ಮಾಡ್ತೀವಿ. ಥರ್ಡ್ ಒನ್ ಇಸ್ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಡಿಸೈನ್. ಆ ಡಿಸೈನ್ ಒಟ್ಟಿಗೆ ಇಮೇಜ್ ಒಟ್ಟಿಗೆ ಈ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಡಿಸೈನ್ ಸಹ ಮಾಡ್ತೀವಿ. ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಡಿಸೈನ್ ಅಲ್ಲಿ ಏನು ಅಂದ್ರೆ ನಮ್ಗೆ ಯಾವ ಮಟೀರಿಯಲ್ ಬೇಕು, ಎಷ್ಟು ಶೇಪ್ ಬೇಕು, ಯಾವ ಶೇಪ್ ಬೇಕು, ಯಾವ ಸೈಜ್ ಬೇಕು, ಅದರ ಒಳಗಡೆ ಸ್ಟ್ರಕ್ಚರ್ ಹೇಗಿರಬೇಕು, ಅದೆಲ್ಲ ರಿಕ್ವೈರ್ಮೆಂಟ್ಸ್ ಅನ್ನ ನೋಡಿ ಮತ್ತೆ ಅದು ಪ್ರಾಪರ್ ಚೆನ್ನಾಗಿದೆಯಲ್ಲ, ಕರೆಕ್ಟ್ ಆಗಿದೆಯಲ್ಲ ಅನ್ನೋದನ್ನ ಎಕ್ಸಾಮಿನ್ ಮಾಡ್ತೀವಿ. ಅದು ಡಿಸೈನ್, ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಡಿಸೈನ್. ನೆಕ್ಸ್ಟ್ ಮಟೀರಿಯಲ್ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಬೇಕಂದ್ರೆ ಬಯೋಕಂಪ್ಯಾಟಿಬಿಲಿಟಿ ಬೇಕು. ಅದು ಯಾವುದೇ ಒಳಗಡೆ ಇಂಪ್ಲಾಂಟ್ ಮಾಡಿದ್ಮೇಲೆ ಮನುಷ್ಯನ ಒಳಗಡೆ ಅದು ಯಾವುದೇ ತರದ ಅಲರ್ಜಿಸ್, ರಿಯಾಕ್ಷನ್ಸ್, ಅಡ್ವರ್ಸ್ ಎಫೆಕ್ಟ್ಸ್ ಅನ್ನ ಮಾಡಬಾರದು. ಸೊ ಆಮೇಲೆ ನಮ್ಮ ಮಟೀರಿಯಲ್ ಏನು ಯೂಸ್ ಮಾಡಿರ್ತಾರಲ್ಲ ಅದು 3 ಫಂಕ್ಷನ್ ಮಾಡಬೇಕು. ಸೆಲ್ ಅಟ್ಯಾಚ್ ಆಗಬೇಕು, ಗ್ರೋತ್ ಆಗಬೇಕು, ಇವೆಂಚುಯಲಿ ಏನಾಗಬೇಕು, ಬೋನ್ ಅನ್ನ ರೀಜನರೇಷನ್ ಅಂದ್ರೆ ಮತ್ತೆ ಹುಟ್ಟಿ ಹಾಕಬೇಕು. So fifth step is 3D printing process. So once the scaffold design and as well as material selection are finalized, so finally now we have finalized the scaffold design and materials are finalized now. Now the actual 3D printing process starts here. Okay, so the chosen technique, whatever the technique it can be. So but here layer by layer it will be done because it is additive techniques. Now the 3D printer precisely deposits or fuses the chosen material following the digital model specification. ಏನೆಲ್ಲ ಡಿಸೈನ್ ಮಾಡಿರ್ತಿಲ್ಲ ಅದೇ ತರ ಪ್ರಿಂಟ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ ಇಲ್ಲಿ, ಅದಕ್ಕೆ ನಾವು 3D printing ಅಂತ ಕರಿತೀವಿ. So next, post processing. Now after printing you will get the post, you need to process first. Why we need this step is to remove certain kind of structures which are not required. Okay, so for example to remove the support structures, cleaning the scaffolds, and as well as performing necessary surface treatments to enhance the biocompatibility and optimize the scaffold's property. ಸೊ ನಾವು ಏನಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತೀವಿ ಪೋಸ್ಟ್ ಪ್ರೊಸೆಸಿಂಗ್ ಅಂದ್ರೆ, ನಮಗೆ ಪ್ರಿಂಟ್ ಆದ್ಮೇಲೆ ಅದರಲ್ಲಿ ಇರುವ ಕೆಲವೊಂದು ಸಪೋರ್ಟ್ ಸ್ಟ್ರಕ್ಚರ್ ಆಗಿ ಯೂಸ್ ಮಾಡಿರ್ತೀವಲ್ಲ, ಅದು ಬೇಡ ನಮ್ಗೀಗ, ಅದನ್ನ ತೆಗೆದು ಹಾಕ್ಬೋದು. ನೆಕ್ಸ್ಟ್ ಕ್ಲೀನಿಂಗ್ ಮಾಡ್ಬೋದು, ಕ್ಲೀನಿಂಗ್ ಇರುತ್ತೆ, ಅದನ್ನ ಸ್ಟೆರಿಲೈಸೇಷನ್ ಅದೆಲ್ಲ ಮಾಡ್ಬೋದು. ಆಮೇಲೆ ನೆಕ್ಸ್ಟ್ ಸರ್ಫೇಸ್ ಟ್ರೀಟ್ಮೆಂಟ್ ಮಾಡ್ಬೇಕಾಗತ್ತೆ, ಅಂದ್ರೆ ಅದನ್ನ ಕರೆಕ್ಟ್ ಆಗಿ ಅದರಲ್ಲಿ ಸರ್ಫೇಸ್ ಏನಾದ್ರು ಇದ್ರೆ ಅದಕ್ಕೆ ಬೇರೆ ಏನಾದ್ರು ಹಾಕಿ ಇದು ಮಾಡಿ ಅದು ಬಯೋಕಂಪ್ಯಾಟಿಬಲ್ ಇದೆಯಾ ಇಲ್ಲ ಅನ್ನೋದನ್ನ ನಾವು ಚೆಕ್ ಮಾಡ್ಬೇಕು. ಇಲ್ಲ ಅಂದ್ರೆ ಅದಕ್ಕೆ ಸ್ವಲ್ಪ ಏನಾದ್ರು ಸರ್ಫೇಸ್ ಅಲ್ಲಿ ಏನಾದ್ರು ನಾವು ಚೇಂಜಸ್ ಮಾಡಿ ಅದನ್ನ ಬಯೋಕಂಪ್ಯಾಟಿಬಲ್ ಆಗಿ ಮಾಡಬೇಕು, ಮತ್ತೆ ಆಪ್ಟಿಮೈಸ್ ಮಾಡಬೇಕು, ಸೊ ದಟ್ ಅದರಲ್ಲಿ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಪ್ರಾಪರ್ಟೀಸ್ ಇರುತ್ತೆ. So that is about the post processing. Next, sterilization. Sterilization comes under make sure that it is the particular bone printed is free from the contaminants. Either it can be the microbe, free from the microbes, free from the any other contaminants, dust or any kind of stuff. So that comes under sterilization. And finally, ready for the clinical use. Make sure that after sterilization, the product should be ready for implanting inside the body. Clinical use andre eno means what you mean by clinical use? To use inside the human being. So it should be ready. After sterilization means it is the final product and there are no contaminants, there are no bacteria. So sterilization ಅಂದ್ರೆ ಅದರಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನೋ ಯಾವುದೇ ತರಹದ ಕಲ್ಮಶಗಳು ಇರಬಾರ್ದು, ಅದೆಲ್ಲ ಕ್ಲೀನ್ ಆಗಿರಬೇಕು, ಅದನ್ನ ಮತ್ತೆ ಮನುಷ್ಯನ ಒಳಗೆ ಹಾಕಕ್ಕೆ ಅದು ಯೋಗ್ಯವಾಗಿರಬೇಕು. Now the 3D printed bone scaffold undergoes sterilization using appropriate methods. Common techniques include autoclaving, ethylene oxide sterilization, or gamma irradiation. So many techniques can be used that can be usually sterilize ಮಾಡಕ್ಕೆ ಬೇರೆ ಎಲ್ಲ ಟೆಕ್ನಿಕ್ಸ್ ಯೂಸ್ ಮಾಡಬಹುದು. These are the techniques which are used for the sterilization. Next, surgical implant. Here I can just give an introduction because you might not be knowing what is autoclaving and all. So autoclave in the sense you are having normal the pressure cooker what you have at your home, no? Similar way in the autoclaving what happens, there will be a temperature, high pressure will be maintained so that any bacteria, any impurities which are there, that will be dead, and the impurities will be made deactivated because of the high temperature and pressure. So here in the particular organs like ear and the liver and all, we can't use it for higher temperature. So we can use for some particular ethylene oxide sterilization sometimes, not always, because hard technique like for bone we can go for autoclaving, not a problem. For since we are studying here bone, we are mentioning it this autoclaving. ಅರ್ಥ ಆಯ್ತಲ್ಲ. ಸೊ ಆಟೋಕ್ಲೇವರ್ ನ ಅಂದ್ರೆ ನಾವು ಪ್ರೆಷರ್ ಕುಕ್ಕರ್ ತರನೇ ಇರುತ್ತೆ, ಅದು ಆಲ್ಮೋಸ್ಟ್ ಹೈ ಟೆಂಪರೇಚರ್ ಹೈ ಇರುತ್ತೆ. ಸೊ ಹಾರ್ಡ್ ಟಿಶ್ಯೂ ಲೈಕ್ ಬೋನ್ ವಾಟ್ ವಿ ಆರ್ ಡಿಸ್ಕಸಿಂಗ್, ಅದಕ್ಕೆ ಆಟೋಕ್ಲೇವ್ ಮಾಡಬಹುದು. ಸೊ ದಟ್ ಅದರಲ್ಲಿ ಮೈಕ್ರೋಬ್ಸ್ ಮತ್ತೆ ಏನಾದ್ರು ಡರ್ಟ್ ಇದ್ರೆ ಎಲ್ಲ ಕಿತ್ಕೊಂಡು ಹೋಗುತ್ತೆ. ಸೊ ethylene oxide sterilization is there. We use the ethylene oxide so that it will sterilize. You just pour like a liquid, it will get sterilized. And or gamma irradiation, you can use the gamma irradiation if any impurities are there, that will be wiped out. Okay. Next, last step is surgical implantation. Now you have printed, now you have post cleaned it, and then you have sterilized it, and finally the surgical implantation. Now you should implant inside the body. Okay, so now it is a bone, now you have to implant inside the body. What you have to do? The surgery should be done. So that's why it is called as surgical implantation of the 3D printed bone scaffold into the patient. Now surgeons carefully position the scaffold in the intended area, so they will ensure that proper alignment and stability is there, so that over time the scaffold provides support for bone regeneration and integrates with the surrounding tissue, so that it will give the bone regenerated and integrates with the tissue. That means after implanting, after surgery you implant it, it should integrate with the other cells like tissues, the muscle structure, so that it will adapt. It should be biocompatible there. Okay, because since you have used the biocompatible materials, it should be biocompatible. Right? There should not be any problem. So that is called, that comes under surgical implantation. So ಇಲ್ಲಿ ಸರ್ಜಿಕಲ್ ಇಂಪ್ಲಾಂಟೇಷನ್ ಏನಂದ್ರೆ, ನಾವು ಈಗ ಬೋನ್ ಅನ್ನ ಒಳಗಡೆ ಹಾಕ್ಬೇಕಂದ್ರೆ ನಾವು ಸರ್ಜರಿ ಮಾಡಲೇಬೇಕಾಗತ್ತೆ. ಸರ್ಜರಿ ಮಾಡೋದು, ಬೋನ್ ಅನ್ನ ಒಳಗಡೆ ಫಿಕ್ಸ್ ಮಾಡೋದು, ಆಮೇಲೆ ಅದನ್ನ ಕ್ಲೋಸ್ ಮಾಡೋದು, ಆಮೇಲೆ ಅಲ್ಲಿ ಅಲೈನ್ಮೆಂಟ್ ಚೆನ್ನಾಗಿ ಕರೆಕ್ಟ್ ಆಗಿದೆಯಾ ಅಲ್ವಾ, ಅದು ಸ್ಟೇಬಲ್ ಆಗಿದೆಯಾ ಅನ್ನೋದನ್ನ ಚೆಕ್ ಮಾಡ್ಕೊಡೋದು. ಆಮೇಲೆ ಅದ್ರ ಈ ಬೋನ್ ಮೇಲೆ ಟಿಶ್ಯೂ ಫಾರ್ಮ್ ಆಗ್ತಾ ಹೋಗುತ್ತಲ್ಲ, ಆ ಟಿಶ್ಯೂ ಫಾರ್ಮ್ ಸರಿಯಾಗಿ ಆಗ್ತಾ ಇದೆಯಾ ಅಲ್ವಾ ಅನ್ನೋದನ್ನ ಚೆಕ್ ಮಾಡ್ತಾ ಇರೋದು. ಅದೆಲ್ಲ ಲಾಸ್ಟ್ ಸರ್ಜಿಕಲ್ implantation process ಅಲ್ಲಿ include ಆಗುತ್ತೆ. So what are the 8 steps? First, you take an image. Second one, digital model generation, make a model. Third, scaffold design. You make a design and then material selected. You have to select the materials what you want to print, like it should be biocompatible on those applications. It should provide cell growth, cell attachment growth, and eventually bone regeneration. And fifth step is what? 3D printing process, which starts here. It depends on whatever the techniques should be used based on the materials, but it should be layer by layer since it is additive approach. Next, post processing. So you need to process it for the after printing it. You need to make sure that there are no other structures which are involved. For example, what we use supports or clean for this thing and all, that should be taken off, and then you clean the scaffold, and then also if any surface modification requires to get the biocompatibility property, that should be done. And then finally you need to sterilize. You can sterilize with many techniques. Sterilize in the sense you need to remove the contaminants. It can be the microbes or any other dirt or any other materials which are present which are not required there. So we can use autoclaving, okay, for high temperature and pH. You can, high temperature and pressure. With the help of that you can use. Then ethylene oxide sterilization, you just pour the ethylene oxide so that it will be sterilized, or you can use the gamma radiation. So last step, so surgical implantation. You take out the do the surgery, keep inside the bone can be implanted inside, and then make sure that proper alignment and stability is there. That's it. And also it should have a supportive environment. That means after inserting the bone, the cells will start growing, or the tissue formation will be there just above that tissue format should support that. So that should be taken care. So that all things comes under this thing. So that was 8 steps under additive manufacturing of 3D printing. This is the first approach. Okay. So next approach is what? Scaffold based techniques in 3D printing of bones. So here we need to have a framework template so that we can go for which helps for the regeneration of the bone tissue. So these techniques involve fabrication of biocompatible and biodegradable scaffolds using 3D printing technology, which can mimic the structure and properties of the natural bone. Again, it is telling the same thing. We are using the 3D printing technology to fabricate biocompatible and biodegradable materials so that it can mimic the structure and property functions of the bone. Next, the scaffold serves as a temporary support structure that provides mechanical stability and guides the growth of new bone tissue. So scaffold we have already discussed. It is well known for mechanical support, so that we are concentrating on the scaffold. That's why it is called a scaffold based techniques. So it offers a 3-dimensional structural framework with interconnected pores that allows for the cell filtration, infiltration, nutrient diffusion, and as well as deposition of extracellular matrix. Now this scaffold, why it is very important is that it has got some interconnected pores inside that, so that it will allow 3 major functions. Cell infiltration will be there. So that means from cells, when they start, they will start moving, right? So that infiltration will be there. For that, pores are required. Next, nutrient diffusion. So the required nutrients for the cell growth should be given, right? From the body we will get it. So those nutrients should come to this particular cells which are growing. For that again we need pores, interconnected pores. Again ECM has that. So next, finally deposition of extracellular matrix. To deposit this extracellular matrix also we need that, so that this particular scaffold will be having in all these 3 properties in 3-dimensional function. So ಇಲ್ಲಿ scaffold ಅಂದ್ರೆ ಇದು 3D printing ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಅಗೇನ್ ಇದು ಫ್ರೇಮ್ವರ್ಕ್ ಅಂತ ಟೆಂಪ್ಲೇಟ್ ತರ ಆಗುತ್ತೆ. ಇದರಲ್ಲಿ ಬಯೋಕಂಪ್ಯಾಟಿಬಲ್ ಇದನ್ನ ಫ್ಯಾಬ್ರಿಕೇಟ್ ಮಾಡೋದು ಬಯೋಕಂಪ್ಯಾಟಿಬಲ್ ಮತ್ತೆ ಬಯೋಡಿಗ್ರೇಡಬಲ್ ಮಟೀರಿಯಲ್ಸ್ ಯೂಸ್ ಮಾಡಿಕೊಂಡು, ಆಮೇಲೆ ಇದು ಮೆಕ್ಯಾನಿಕಲ್ ಸ್ಟೆಬಿಲಿಟಿಗೆ ನಾವು ಯೂಸ್ ಮಾಡ್ತೀವಿ ಅಂತ ಪ್ರೀವಿಯಸ್ ಕ್ಲಾಸ್ ಅಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಿದ್ದೇವೆ. ಆಮೇಲೆ ಇದರಲ್ಲಿ ಇಂಟರ್ಕನೆಕ್ಟೆಡ್ ಪೋರ್ಸ್ ಇರುತ್ತೆ. ಆ ಪೋರ್ಸ್ಗಳು ಸೆಲ್ ಇನ್ಫಿಲ್ಟ್ರೇಷನ್, ಸೆಲ್ ಮೂವ್ಮೆಂಟ್ ಆಗ್ತಾರೆ, ಗ್ರೋ ಆಗ್ಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಆಮೇಲೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಏನೆಲ್ಲ ಸೆಲ್ಗೆ ಗ್ರೋತ್ ಆಗಲಿಕ್ಕೆ ಬೇಕು ಅದನ್ನ ಪ್ರೊವೈಡ್ ಮಾಡ್ಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಮತ್ತೆ ಈ ಎಕ್ಸ್ಟ್ರಾ ಸೆಲ್ಯುಲರ್ ಮ್ಯಾಟ್ರಿಕ್ಸ್ ನಾನು ನಿನ್ನೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ವಲ್ಲ, ಅದನ್ನ ಡೆಪಾಸಿಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಸಹ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. So steps involved in the scaffold based 3D printing of bone. The first thing is here design. First we should have a design in the scaffold, 3-dimensional structure design. So what we are doing, a digital model of the desired bone structure you need to prepare by using what? CAD software. So what are the parameters? We need to see whether the shape is is customized, or whatever the shape is there, what we need, whether it is there, and then size, okay, or pore architecture and mechanical properties. So all these properties should be checked in the design. Okay, design ಅಲ್ಲಿ ನಾವು CAD ಸಾಫ್ಟ್ವೇರ್ ಯೂಸ್ ಮಾಡಿ ಅದು ಹೇಗೆ ಯಾವ ಆರ್ಗನ್ ಹೇಗೆ ಇರಬೇಕು ಆ ಸ್ಟ್ರಕ್ಚರ್ ಅನ್ನು ಡಿಸೈಡ್ ಮಾಡ್ತೀವಿ. ಅದರಲ್ಲಿ ಶೇಪ್ ಹೇಗಿರಬೇಕು, ಸೈಜ್ ಹೇಗಿರಬೇಕು, ಅದರ ಒಳಗಡೆ ಇರುವ ಪೋರ್ಸ್ ಅಂದ್ರೆ ಹೋಲ್ಗಳು, ಅದು ಪೋರ್ಸ್ ಅದು ಎಷ್ಟು ಹೇಗಿರಬೇಕು, ಅದು ಅದರದ್ದು ಡೈಮೆನ್ಷನ್ ಸೆಟ್ಟು, ಆಮೇಲೆ ಅದರದ್ದು ಮೆಕ್ಯಾನಿಕಲ್ ಪ್ರಾಪರ್ಟಿ ಎಷ್ಟು ಬೇಕು, ಎಲ್ಲ ಡಿಸೈನ್ ಅಲ್ಲಿ ಮೆನ್ಷನ್ ಆಗಿರುತ್ತೆ. ಸೊ ನೆಕ್ಸ್ಟ್ ಮಟೀರಿಯಲ್ ಸೆಲೆಕ್ಷನ್. Obviously you need to have biocompatible or biodegradable material should be chosen. Yesterday we discussed about these materials, PCL, PLGA and natural polymers like collagen and gelatin. Same thing, copy paste here, nothing else. The third thing, material select ಅಂದ್ರೆ just ನೀವು biodegradable biopolymers ಅನ್ನು ಯೂಸ್ ಮಾಡಬೇಕು. ನಿನ್ನೆ ನಾವು ಹೇಳಿದಾಗ ಹಾಗೆ PCL, PLGA ಅದನ್ನ ಎಕ್ಸಾಂಪಲ್ಸ್ ಅಂತ ಕೊಡಬಹುದು. Next 3D printing process. Now 3D printing process we have been studying, this is the third time we are going to study. So here 3D printing process, again it begins with the loading of the selected materials into the 3D printer. That whatever the materials we need to print, that should be loaded in 3D printer. Now the printer then deposits or solidifies the material layer by layer. So following the digital design, it will just follow this particular, whatever the design is there, it will keep printing. Now the printing technology can vary, including extrusion methods, inkjet method and stereolithography. All these three methods I've discussed. If you have not understood properly, you just go back to those videos, see those videos, understand and come back here and see. Next, that was about 3D printing process. Next, pore formation. Now pore need to be formed, right? So during the printing process, what happens? Scaffold is designed to have a porous structure. Now this particular scaffold will have a design structure for the porous structure. That means the structure should have number of pores. Wherever that interconnected pores should be there, which are required for this particular cell mobility and as well as nutrient transportation. So these pores provide a space for the self-infiltration, nutrient supply and vascularization. Self-infiltration we discussed, nutrient supply we discussed, vascularization, that is like to form the blood vesicles or tubes kind of structures. So that is why we require. So various techniques can be used here. Again here we can have a control over pore size, distribution and interconnectivity. ಸೊ ಇಲ್ಲಿ ಪೋರ್ ಫಾರ್ಮೇಶನ್ ಅಂದ್ರೆ ಹೋಲ್ ಕ್ರಿಯೇಟ್ ಮಾಡೋದು. ಆ ಹೋಲ್ ಕ್ರಿಯೇಟ್ ಏನಕ್ಕೆ ಮಾಡ್ತಿದೀವಿ ಅಂದ್ರೆ ಅಲ್ಲಿ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಸಪ್ಲೈ ಆಗಬೇಕು, ಸೆಲ್ ಇನ್ಫಿಲ್ಟ್ರೇಷನ್ ಆಗಬೇಕು, ಮತ್ತೆ ವ್ಯಾಸ್ಕುಲರೈಸೇಷನ್ ಅಂದ್ರೆ ಈ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಫಾರ್ಮ್ ಆಗಬೇಕು. ಯಾಕಂದ್ರೆ ನಮ್ಮ ಬ್ಲಡ್ ಕನೆಕ್ಷನ್ ಇರಬೇಕಲ್ಲ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಸಪ್ಲೈಗೆ ಸಹ. ಸೊ ದಟ್ಸ್ ಹೌ ಇಟ್ ವರ್ಕ್ಸ್. ಸೊ ದಟ್ ಇಸ್ ಅಬೌಟ್ ಪೋರ್ ಫಾರ್ಮೇಶನ್. ನೆಕ್ಸ್ಟ್ ಫಿಫ್ತ್ ಪಾಯಿಂಟ್ ಇಸ್ post processing. Now you have got the 3D final product. Post processing, again here whatever this thing is there, why we use to remove the unwanted materials. So here supportive, what are the unwanted materials? Support structures. Okay, for example, if I use this as a support structure to print this particular 3D, imagine these things as a 3D ear here. Now I want only this. Now the supporting material should be cut off. Now I will be taking it off. ಅರ್ಥ ಆಯ್ತಲ್ಲ. ಸೊ ಇವಾಗ ನನಗೆ ಇದನ್ನು ಮಾತ್ರ ಪ್ರಿಂಟ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಇದು ಸಪೋರ್ಟಿವ್ ಮಟೀರಿಯಲ್ ಆಗಿ ಯೂಸ್ ಮಾಡಿರ್ತೀನಿ. ನಾನು ಈ ಸಪೋರ್ಟ್ ಮಟೀರಿಯಲ್ ಅನ್ನ ತೆಗೆದಾಕ್ತೀನಿ, ನಂಗೆ ಇದು ಮಾತ್ರ ಉಳಿಯುತ್ತೆ. ಸೊ ಅದು ಆಫ್ಟರ್ ದ ಪ್ರೊಡಕ್ಷನ್. ಸೊ ಅದು ದಟ್ ಕಮ್ಸ್ ಅಂಡರ್ ಪೋಸ್ಟ್ ಪ್ರೊಸೆಸಿಂಗ್. ನೆಕ್ಸ್ಟ್ ಸ್ಟೆರಿಲೈಸೇಷನ್, ಅಗೇನ್ ಬೈ ಯೂಸಿಂಗ್ ಆಟೋಕ್ಲೇವ್, whatever we discussed in the previous additive listening. And next, surface treatments to enhance the biocompatibility. Now post processing ಆದ್ಮೇಲೆ next step we are having cell seeding and culture. So now cells we need to seed. That means now after this, now we have got a scaffold, right? Now we need to seed cells inside that, so that now whatever the cells, for example mesenchymal cells or osteoblast cells. Why we are talking about mesenchymal cell and osteoblast cells? Because these are the major components of the cells presence in the bone. Now we are preparing bone, right? So mesenchymal cells, osteoblasts, that can be added as inside the extracellular matrix or scaffold. Now scaffold is there, ಅದರೊಳಗಡೆ ನಾವು ಏನ್ ಬೆಳಿಬೇಕು, ಆ ಸೆಲ್ಸ್ ಬೆಳಿಬೇಕು. ಆ ಸೆಲ್ಸ್ ಯಾವುದು? ಮೆಸೆಂಕೈಮಲ್ ಸೆಲ್ಸ್, ಓಸ್ಟಿಯೋಬ್ಲಾಸ್ಟ್ ಸೆಲ್ಸ್. ಇವೆ ಯಾಕೆ? ಯಾಕಂದ್ರೆ ದೇ ಆರ್ ದ ಬೋನ್ ಅಲ್ಲಿ ಇರುವಂತ ಸೆಲ್ಸ್ ಗಳು ಇವೆ. ಸೊ ಈ ಸೆಲ್ಸ್ ಗ್ರೋ ಆಗಬೇಕು. ಅದನ್ನ ನಾವು ಸ್ಕ್ಯಾಫೋಲ್ಡ್ಸ್ ನಾವು ಈಗ ರೆಡಿ ಆಗಿದೆ. ಅದರೊಳಗಡೆ ಸೀಡಿಂಗ್ ಅಂದ್ರೆ ನಾವು ಈಗ ಬಿತ್ತನೆ ಅಂತ ಹೇಳ್ತೀವಲ್ಲ ಆ ತರ ಅದನ್ನ ನಾವು ಹಾಕ್ತೀವಿ. ಸೊ ದಟ್ ದಿಸ್ ವಿಲ್ ಸೀಡೆಡ್ ಅಪ್. ಸೊ ದಟ್ ನಾವು ಸೀಡೆಡ್ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಇಸ್ ದೆನ್ cultured under the appropriate condition to promote the cell attachment, proliferation and finally the formation of new tissue within a scaffold. Now this seeded scaffold. Now we have seeded these things to grow. So now to grow, what are the environment is required? That should be maintained. So that is called as appropriate condition is maintained, so that cell attachment will happen. Cell attachment is why you required, because it should grow, right, for implantation and all. So cell attachment should be there. Next, proliferation will be there. Next, proliferation in the sense cells will start developing and eventually they will growing to form a tissue. And finally, formation of the bone tissue within the scaffold. Now we are having a bone tissue within the scaffold. Now finally, final step is implant it. So where you are going to implant it? So again, here one of the major steps is to make sure that this particular once the scaffold construction is done, so it should make sure that that is sufficient maturation. That means correct it is developed. So that means it has developed properly. So it can be implanted into the patient's body, so that there should not be any adverse effects inside the body. So now the scaffold provides structural support while the surrounding cells and the blood vessels infiltrate and replace the scaffold with newly formed bone tissues, so that first it will give the structural support for all the seeded cells to grow. Once it starts growing, the surrounding cells will start growing, blood vessels will keep forming, and then finally eventually it will replace the scaffold with newly formed bone tissue. Now the scaffold is biodegradable, that will go off and the new formed tissue will come up. So over time the scaffold degrades. It degrades, leaving behind the functional regenerated bone. Only bone will remain. So we are using the scaffold to grow the bone cells. Once the bone cells starts growing, it forms a bone and scaffolds will start degrading. Okay, understood. So finally we can have that. So implantation alli enu andre ನಾವು ಬೋನ್ ಇರುತ್ತಲ್ಲ ಆ ಬೋನ್ ಅನ್ನ ಸಫಿಷಿಯಂಟ್ ಮ್ಯಾಚುರೇಷನ್ ಆಗಿದೆಯಲ್ಲ ಅಂತ ನೋಡ್ಕೋಬೇಕು. ದಿ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಅನ್ನ ನಾವು ಈಗ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಅನ್ನ ನಾವು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ಮೇಲೆ ಆ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಒಳಗಡೆ ಸೆಲ್ಸ್ ಇರುತ್ತೆ. ಯಾವ ಸೆಲ್ಸ್ ಇರುತ್ತೆ? ಮಿಸೆಂಕೈಮಲ್ ಅಂಡ್ ಓಸ್ಟಿಯೋಬ್ಲಾಸ್ಟ್ ಸೆಲ್ಸ್. ಸೊ ಈ ಸೆಲ್ಸ್ ಗ್ರೋ ಆಗಿ ಟಿಶ್ಯೂ ಆಗಿ ಬೋನ್ ಆಗಿ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ಇವೆಲ್ಲ ಆಗ್ತಿದ್ದಂಗೆನೆ ಈ ಏನು ಹೊರಗಡೆ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಆಗಿರೋದಲ್ಲ 3D ಪ್ರಿಂಟ್ ನಾವು ಫಸ್ಟ್ ಡಿಸೈನ್ ಮಾಡಿರ್ತೇವೆ, ಅದು ಡಿಗ್ರೇಡ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಅಂದ್ರೆ ಅದು ಅಲ್ಲಿ ಇದು ಕಟ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಅದರಿಂದ ನಮ್ಗೆ ಲಾಸ್ಟ್ ಗೆ ಸಿಗೋದೇನು? ರೀಜನರೇಟೆಡ್ ಬೋನ್. ಅರ್ಥ ಆಯ್ತಾ? So that's it. The materials used for the 3D printing bones are, we are having the many, depending on the many specific 3D techniques, we are having different materials. The biocompatible polymers, ceramics, scaffolds and cell embedded materials. So biocompatible polymers, as we discussed in the previous topic, it should be compatible with the human tissues, do not cause any adverse effects. Example, polyethylene, polycaprolactone and polylactide, and as well as polyvinyl alcohol. These all comes under biocompatible polymers as an example. So ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಇವೆಲ್ಲ ಅಡ್ವರ್ಸ್ ಎಫೆಕ್ಟ್ ಆಗಬಾರ್ದು ಬಾಡಿಯಲ್ಲಿ. ಆ ತರ ಪ್ರಾಪರ್ಟಿ ಇದ್ರೆ ಅದಕ್ಕೆ ಬಯೋಕಂಪ್ಯಾಟಿಬಲ್ ಮಟೀರಿಯಲ್ಸ್ ಹೇಳ್ತಾರೆ. ಎಕ್ಸಾಂಪಲ್ಸ್ ಇಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾರೆ. ನೆಕ್ಸ್ಟ್ ಯು ಕ್ಯಾನ್ ಸಿರಾಮಿಕ್ಸ್. ಸೊ ಸಿರಾಮಿಕ್ಸ್, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಹೈಡ್ರಾಕ್ಸಿ ಅಪಾಟೈಟ್. Hydroxy apatite is one of the biocompatible ceramic material. So that has been shown an effective ceramic material for the 3D printing of bones. It is a very effective material which will act as a biocompatible material. Example, hydroxyapatite, calcium itself, and then calcium phosphate. You are having a tricalcium phosphate also. Why calcium here? Because bone also contains the calcium, which is again major component. That's why not only the polymers, we should have the ceramics also, which will give you the calcium content for the bone. So ಇಲ್ಲಿ calcium ceramics ಏನಕ್ಕೆಪ್ಪ ಅಂದ್ರೆ ಬೋನ್ ಅಲ್ಲಿ ಜಾಸ್ತಿ ಇರೋದು ಕ್ಯಾಲ್ಸಿಯಂ. ಸೊ ಅಲಾಂಗ್ ದ ಪಾಲಿಮರ್ ಒಟ್ಟಿಗೆ, ಪಾಲಿಮರ್ ಬಯೋಮೆಟೀರಿಯಲ್ಸ್ ಒಟ್ಟಿಗೆ ನಮಗೆ ಸಿರಾಮಿಕ್ಸ್ ಅನ್ನು ನಾವು ಆಡ್ ಮಾಡಬೇಕಾಗುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ಸ್. ಸೊ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ಸ್ we discussed, it will provide a supportive framework for the cell growth and as well as to develop. In the case of 3D printing of the bones, scaffolds can be used to create a specific shapes or structure of the bone tissues or grow around. So examples, we are having PGA, PLLA and PET. So these are the example for the scaffolds. So scaffolds ಏನಕ್ಕೆ ಬೇಕಂದ್ರೆ ಇದು ಸಪೋರ್ಟ್ ಕೊಡುತ್ತೆ. ಏನಕ್ಕೆ? ನಮಗೆ ಬೋನ್ ಫಾರ್ಮ್ ಮಾಡಕ್ಕೆ. ಇದು ಇರಲ್ಲ ಅಲ್ಲಿ ಸ್ಕ್ಯಾಫೋಲ್ಡ್, ಅದು ಬೋನ್ ಫಾರ್ಮ್ ಆಗೋವರೆಗೂ ಇರುತ್ತೆ. ಬೋನ್ ಫಾರ್ಮ್ ಆಗ್ತಾನೆ ಡಿಗ್ರೇಡ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಆಯ್ತಾ? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇವಾಗ ನಿಮ್ ಸ್ಟಿಚ್ ಎಲ್ಲ ಮಾಡಿರ್ತಾರೆ, ಅದು ತುಂಬಾ ದಿನ ಆದ್ಮೇಲೆ ಅದು ಅದ್ರ ಪಾಡಿಗೆ ಅದೇ ಹೋಗ್ತಾ ಇರುತ್ತಲ್ಲ, ಅದಕ್ಕೆ ಡಿಗ್ರೇಡೇಷನ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಸೊ ಆವಾಗ ನೀವು ಅದು ಸ್ಟಿಚ್ ಇದು ಮಸಲ್ ಎಲ್ಲ ಕೂಡ್ಕೊಂಡು ಬಂದಾಗೆ ಅದು ಸ್ಟಿಚ್ ಹೋಗ್ತಾ ಇರುತ್ತೆ. ಹಾಗೆ ಸ್ಕ್ಯಾಫೋಲ್ಡ್ ಸಹ. ಸೊ ಅದು ರೀಜನರೇಟೆಡ್ ಬೋನ್ ಆದಂಗೆ ಇದಾಗ್ತಾ ಅದು ಡಿಗ್ರೇಡ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಎಕ್ಸಾಂಪಲ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಸೊ ಎಕ್ಸಾಂಪಲ್ PET, PLLA and PGA. So next is cell embedded materials. Cell embedded materials are nothing but materials that contain living cells. Cell embedded in the sense cells are inserted inside this particular material. So cells are mixed with the materials. So cells materials ಒಳಗೆ mix ಆಗಿದೆ, ಅದಕ್ಕೆ ನಾವು cell embedded materials ಅಂತ ಕರೀತೇವೆ. So here cells should be living, so which can be used to seed the 3D printing structure. Now why we use these particular cell embedded materials? For the printing, 3D printing of structure. Okay, so now the cell then grow, develop into the functional bones tissues over time. Examples, we are having gelatin, methacrylate, and we are having alginate. So cell embedded andre materials ಒಟ್ಟಿಗೆ living cells ಇರುತ್ತೆ, ಆ living cells grow ಆಗ್ತಾ ಹೋಗಿ ಲಾಸ್ಟ್ ಗೆ ನಿಮಗೆ ಟಿಶ್ಯೂ ಅನ್ನು ಕೊಡುತ್ತೆ. So